Hi, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Trine Grønhaug Halvorsen. Listeners, Trine is professor in the section for Pharmaceutical Chemistry within the Department of Pharmacy at the University of Oslo in Norway. And today we are looking forward to discussing her career and some of her research on dried blood spot sampling and detecting peptides in anti-doping samples. So, Trine, welcome to the show today. How are you?
Thank you. I'm very excited to be here. It's a new thing to me. So, I'm looking forward to this conversation.
Well, we are excited to have you with us, and we're looking forward to learning more about you and the wonderful work that you do. But perhaps you could start by telling us a little bit more about yourself. Trine, how did you initially get interested in anti-doping science and find your way to your current position there at the University of Oslo?
Well, I have always been interested in sports. As a child, I was mainly watching it at TV, but I always found it very interesting. And of course, from time to time, you had this news about doping scandals, but I didn't really think so much about it. But my really first interest in the anti-doping science, I guess, was during my master's year. I'm a trained pharmacist. That's my education. And we have a master thesis. And during that master thesis, I took a course in pharmaceutical bioanalysis. There we had a lecturer from the Norwegian Laboratory of Doping Control, and he was very inspiring. And then after that, I finished my master and I got really into research and wanted to continue. And I was then very lucky and got the opportunity to continue with a PhD at the Department of Pharmacy at the University of Oslo, where I did my education. So I did that, worked with analytical chemistry or pharmaceutical analytical chemistry, and finished a PhD in quite a while ago now, in 2003. Worked a bit in the pharmaceutical industry, a couple of years and a couple of kids. And then I returned to the University of Oslo as a postdoc in 2008. And I've been there since that, first as a postdoc then, and then an associate professor, and now I am a full professor. And still working in the field of pharmaceutical analysis, but with a bit different research profile.
Oh, very cool. Well, thank you for sharing a little bit more about your history and your career path. And you mentioned that you are now a full professor there at the University of Oslo. Can you give us some insight into what do you do in your current role, and perhaps what is the scope of the research that you do?
In my current role as a full professor at the University of Oslo, you have a combined research and teaching position. So I do research and teaching 50-50. And for teaching, just briefly about that, I teach the pharmacy students at bachelor and master's level in pharmaceutical analytical chemistry. So it's like quality control of active drug substance and drug products, and also bioanalysis. So we teach them both analytical techniques and applications. And then for research, I'm into the development of strategies and technologies to improve and simplify determination of proteins and peptides that are biological relevant, like biomarkers, drugs, or doping agents from biological matrices. And I do that by liquid chromatography mass spectrometry, or LCMS. And this has, in a way, been my research interest since I started then as a postdoc in the lab that I am now. And we have been, maybe not since I started in 2008, but for a long time we have been interested in some of the biomarkers that we have been working on are also on the doping list. So we have been working a lot with human chorionic gonadotropin, which is a biomarker for pregnancy, but also for cancers, and it is on the doping list. And it is a very challenging analyte. You can do a lot of fun research with this analyte. So it's been following me since then. And then we started with dried blood spot sampling or microsampling for proteins. And for the last 5, maybe 7 years, we have been moving more towards like still working on microsampling, but we try to integrate some of the sample preparation that you do when you do protein analysis into this sampling unit. And we have developed something that we are called smart sampling, where we do this. And I think the background is this interest in dried blood spot sampling and microsampling. And currently there is this move to get sampling of, like when you do patient samples, you want to move it out of the hospital and to where the patient and caretaker can take it themselves. And that's kind of similar as for the doping analysis.
Very interesting. So it sounds like your research has both clinical and anti-doping relevance, which I think is phenomenal. And it sounds like you've been able to maybe see some advances and changes in the field over time. So if you had to look at the landscape today, Trine, what are some of the current challenges or maybe limitations of dried blood spot sampling in the context of anti-doping specifically?
I think that in the context of anti-doping, one big challenge is at the moment that there are few analytes that you can analyze from dry blood spots. Dry blood spots has a huge potential because it's so simple. The sampling technique and the samples that you take, they are small, so the storage and everything is simple with dry blood spots. But at the moment, there are too few analytes that routinely can be tested for anti-doping. So as a result, the dry blood spots are used, well, at least many places, it's used in addition to and not instead of the other ways of sampling, like sampling blood and urine. And maybe instead of making it easier for the athlete, then it's an additional sample. And the reason for this, that you still have a rather limited analytical menu, I think, is that when you move from regular blood samples to dry blood spots, you get a much lower sample volume. And this becomes especially challenging for the lower band analytes. And then it can be difficult also to extract analytes out of the dry blood spots. So there are these things that makes the dry blood spots a bit more challenging than the regular blood samples. Maybe just because we have less experience with it, but they have to be overcome for dry blood spots to be fully implementable in anti-doping context.
That makes sense, Trine, and I appreciate you giving us an overview. And we mentioned earlier specifically that you're interested in peptides, and let's talk a little bit more about peptide doping agents. Can you tell us what are some examples of some peptide doping agents and maybe what makes them particularly difficult to detect and quantify in anti-doping samples?
What we call peptide doping agents, that is the doping agents that are built from amino acids, and they can vary a lot in size. They can be quite small, maybe up to 10 amino acids, and that's the agent that are called gonadotropin-releasing hormone analogs like buserelin or leuprolide and triptorelin. Or they can be quite big, like insulin, 55 amino acids. And in fact, they can even be bigger, but then we normally call them proteins. And the challenge with these peptide doping agents, they can be synthetic. That's maybe not a challenge, but they can also be endogenous, like similar to what you have in the body yourself. And then if they are that, then they can become quite difficult to monitor because it's difficult to differentiate it between like what do your body produce or what have you been taking as an external agent. So that's one challenge with the peptides as a doping agent. In addition, some of them have very short half-life, so there is a very limited time when you can monitor them, and some are present at very low concentrations. So that's challenging from if you take them as a doping agent, and also there are some analytical challenges that can make it complicated to monitor some of the peptides at least.
Definitely. I think you're simplifying it, certainly, in terms of the complications analytically. Perhaps we can get into some of those details later. But you introduced this idea of smart sampling earlier in talking about your research. So, Trine, can you maybe explain the concept of smart sampling for our listeners who may not have heard this term before and how it could potentially be applied in anti-doping for dried blood spot samples?
So smart sampling is what we call our research field. And what we do in smart sampling is that we integrate sample preparation that is normally performed when the sample arrives to the lab into the sampling step. So normally, if you use dry blood spots as a sampling technique, then you take the sample, you let it dry, and then the sample is transported to the lab for analysis. And when the sample arrives in the lab, you have to do some preparation of the sample before you can do the analysis. And this sample preparation can be quite time-consuming and laborious. So with smart sampling, we try to move some of this sample preparation so that we integrate it into the sampler. So when you apply the sample to the right blood spot card, then the sample preparation starts. The idea is then that when the sample is dry and transported to the lab, and the sample gets to the lab, then you can only do a very easy or simple extraction of the analyte out of this dry blood spot card and the analysis. So that it's kind of a simplification of the analytical procedure. And up to now, we have worked with integration of two different sample preparation steps. One is affinity cleanup. Then we call this sampler for smart affinity samplers. And that is a technique that's very advantageous if you have low abundant analytes, like analytes that are present in low concentrations in the body. Because what we do then is that we kind of fish out the analyte and it selectively bind to the paper and we can wash off all the other components. The other sample preparation step that we have included in the paper in these smart samplers are proteolysis. And then we call the samplers smart proteolysis samplers. And proteolysis is a sample preparation technique that is done when we have proteins. And proteins are then these large peptides, and we can proteolyze them, divide them into smaller peptides before analysis. And then this is very beneficial for the analysis afterward. If you want to do a quantitative analysis of proteins by liquid chromatography, mass spectrometry, then you normally do this proteolysis step. And it can take up to 16 hours to do that. So that is what we try to integrate in the samplers. And I think that both these affinity samplers and the proteolysis samplers can have advantages in anti-doping. The affinity samplers will be of interest if you have analytes that are present in low amounts in blood. It is already used in the lab, but then after the sample has arrived. But if you then can do it to move it into the dry blood spots, then it could be a much more simple sample preparation when the sample arrives in the lab. And it could be used for proteins and peptide, but it can also be used for other analytes which are present in low abundance. And you have antibodies available that can selectively fish out your analyte of interest. The other samplers, the proteolysis samplers, I also think can be of interest, even just by directly if you have high abundance proteins that you would like to monitor, or also if you monitor this at lead biological passport. But this is not something that we have explored yet, so it's something that could be explored.
Definitely. Trine, well, this sounds fascinating, and it sounds like it has a lot of potential to increase efficiency in the labs because some of this work is being done already when the sample's collected. So to kind of streamline the process in anti-doping labs. So let's talk then next about some of your recent research and the papers that you and your group have been putting out there. Last year, you published a review article along with Leon Rubset examining advances in protein determination from dried matrix spots, including these dried blood spots that we've been talking about. So, Trine, can you share some of the key advances that have really occurred in recent years that you think could impact anti-doping?
I think that can have a big impact in the anti-doping and the use of dry blood spots is that we get more and more sensitive instrumentation, like LCM instrumentation. And it's more sensitive and can give us better differentiation between the masses. And this will impact the dry blood spot analysis by just making it possible to determine more doping agent from dry blood spots because you can monitor lower concentration levels. So I think that is one advance that have occurred. And it is a race between the vendors to develop better instrumentation all the time. So that can have an impact. I also think that there is a lot of companies now that develop new microsampling technologies. There it is technologies that simplifies the sampling. For instance, this Tasso device that you can put on the upper arm. And there are other devices that makes it possible to collect plasma. They do a separation of the blood that is collected into plasma during the sampling. And both of these improvements, and maybe others that are going on as well, they will have an impact in anti-doping. And maybe also there will be this possibility of collecting larger volumes than you can do now. And that will also make it more easy.
Definitely. And I know the dried blood spots are relatively small, specifically addressing this idea of potentially being able to collect a larger volume. Is there a limitation to how much you can actually collect with a dried blood spot? Like, could we make the spots larger?
I guess you can make the spots larger, but at the moment you typically collect up to 30 microliter per spot, depending on the size of the spot. And there will be this crossing point, I think, where because you're collecting capillary blood, and if you try to draw too much blood at once, I think there is a risk that you also get maybe some of the leakage of the liquid from tissue around and not only the capillary blood. But I don't really have any numbers about that. I also think that some of the things that we do in our research, it may have an impact in anti-doping, but it's still in the research phase. So it will be a much larger time frame before we see a potential implementation.
Very cool. Well, Trine, it sounds like there's been some great work done in this area, particularly in recent years, but I think there are a lot of areas of opportunity where research is still being conducted, including some of the work that you're doing. So what do you see as some of the biggest areas of opportunity in this research area?
Well, the simplicity of the dry blood spots makes it very appealing in doping context for both the athletes and the doping control officers because the sampling is so easy. I briefly maybe mentioned it earlier, but I think that if you can increase the analysis manual, like the number of analytes that you can determine, then that is a great research opportunity if you want to make the technique more useful in anti-doping research. And first, when this is in place, it will be fully valuable. Because if the athlete must take both urine, blood, and dry blood spot sample, then it's just adding stress on the athlete and not reducing it. Another thing may be this athlete biological passport, like extended use of dry blood spot to monitor. That will also be very valuable because then maybe you need to take more samples and then the sampling method should be simple and user-friendly.
That makes sense. I think you brought up some really good points here. And I really like that you highlighted just the burden on the athlete. If we keep kind of adding these new technologies and tests, we're not actually making it easier for the athletes. So I think that is an important thing for us scientists to keep in mind. Let's talk a little bit more about some of your research. So I know in another recent study, you and some of your colleagues investigated the use of the smart proteolytic samplers that we talked about earlier for quantifying proteins in biological matrices using liquid chromatography mass spectrometry. So Trine, can you give us the background about why this study was important and also how it was conducted?
The first proteolytic samplers we described some years ago, but mainly the focus has been on, does it work? Is the sample digested? And that is, of course, important that it is. But for these samplers to be truly useful in a realistic setting or on real samples, the production or the proteolysis of the protein into peptides needs to be repeatable so that you get reliable results when you drip your sample onto the sample. So in this study, we wanted to assess this a bit more, looking into how the samplers performed quantitatively, both from simple solutions containing standard model proteins and also to more complex biological matrices. So it was a kind of a combined study where we first did some evaluation with the simple model samples to see how does the paper quality affect the digestion of proteolysis and how does the chemistry we use to covalently link our digestin enzyme to the samplers affect the results. So that was the initial part. And then we did a quantitative assessment like linearity and repeatability for both this simple matrix and the complex matrix. And with a complex matrix, I mean a serum that we have diluted a bit.
That makes sense. And then perhaps you could go into some of the results in terms of the reproducibility or repeatability, as well as what the results that you found mean for the field of anti-doping.
When we looked into this quantitative assessment, then we saw that if you use this very simple buffered solution, it works quite well. So it gives us a satisfactory linearity and also the repeatability of the peptides that are produced. They are quite good. So one of the functionalization techniques was better, but still it was quite good. But then when we moved into the more complex biological matrix, like the serum sample, the repeatability or reproducibility seemed to be more peptide dependent. And we could see that doing this quantitative assessment without any form for correction, like either internal correction with correcting two peptides, like we have one peptide in a protein and then correct the amount with another peptide from the same protein, then it didn't really work. It was a lot of variation. So I think that these samplers, you can get reliable quantification, but you need to do more work and optimization for these proteolysis samplers to be really useful in quantitative studies. Maybe you don't have to optimize the samplers, but you need to implement the use of internal protein, internal standards, or some kind of correction to get results that you can trust. And I think one of the reasons for this is that when you take these samples and you leave them for drying, and this is done in room temperature, and when you do things in room temperature, then the temperature can vary, the humidity can vary, so you introduce quite a lot of variation in the sample. So some kind of internal correction needs to be done for this to be very good in ways of quantitative. So it has a potential, but more work is needed.
That makes sense. Kind of needing some sort of way to account for the environmental conditions when the sample was collected and dried. So it sounds like more research is needed in this particular area before it's ready for prime time and anti-doping. But I think you and your colleagues are moving things down the pipeline. You recently published a new study that took the important step of moving from kind of like a proof of concept to fully validating the utility of smart samplers for quantitative analysis of human chorionic gonadotropin, or HCG, in biological matrices. So Trine, can you talk a little bit about this study as well and tell us a little bit more about how you conducted it?
In this study, we do a full method validation according to the European Medicine Agency's guidelines on how to do method validation of bioanalytical methods. The goal was to investigate and then hopefully demonstrate that these smart affinity samplers, so the samplers that can then fish out the analyte that you want to monitor, to see how they work in quantitative analysis. And we used then a human chorionic gonadotropin as a model analyte. And that was the same analyte as we had been working on when we had a proof of concept study. So we had some indications that the methodology was robust, working, like giving repeatable data, but we hadn't really done a systematic evaluation of it. And I have to say that the samplers, we make them in-house, so it's not the commercial product that is available. So the thought was that if we were able to do this validation, even though we make them ourselves, like in-house, then it gives a good indication that this is a procedure that is very robust. So in the study that you mentioned, we first did some fine-tuning of the original proof of concept method to optimize some parameters. And then we did a comprehensive validation, which was then guided by the European Medicines Agency's guideline on bioanalytical method validation. And what we looked into then was selectivity of the method, calibration curves, ranges, accuracy and precision, and matrix effects and carryover. So we looked into all these and everything was good, I think.
Definitely. And this study specifically looked at HCG, which we mentioned earlier in our conversation. Can you explain for listeners who might not be familiar with HCG, why is this particularly relevant for anti-doping?
So HCG is a glycoprotein hormone that is normally produced during pregnancy. So it's produced in large amount during pregnancy. But in doping context, it is on the doping list for male athletes. And that is because if a male athlete takes testosterone or anabolic steroids, then the body's own production of testosterone is downregulated. So the body produces less testosterone. But if you then take HCG or human chorionic gonadotropin, then you can counteract this effect and the body continues to produce testosterone even if you take the anabolic steroids.
That makes sense. So then let's talk about these particular findings where you were using this smart sampler for HCG. What were the findings of this study? And maybe looking at the broader picture, why is it important for anti-doping?
We think that these affinity samplers where we fish out the analytes that we're interested in can have potential, especially for some of the low abundant doping agents, a protein or peptide doping agent maybe in particular. And we know that in order for a method to be used, it needs to be robust. It needs to have good quantitative performance. Well, that is if we want to use it in doping analysis or in regulated bioanalysis. So that was why we wanted to investigate it. And then, of course, if we want to use a method like this in doping analysis, then we also need to see how this method compares to the methods that are used in doping analysis at the moment. So we are planning to, or we have almost finished a study where we do a comparison study with the routine immunological method that is used by the Norwegian Laboratory for Doping Control in analysis of HCG from the regular dried, not blood spots, but serum spots. So we put on serum as a matrix and not whole blood. Our goal, what we really wanted to see was if we could make this confirmation that the method works, if we could do that. And I think that this is kind of the next step for the method or the concept to move forward, to be applied in a real setting.
I think that is remarkable. And it sounds like it is an exciting time for this particular line of research. But Trine, I know you're working on a lot of different things at the moment. Is there another project that you're working on now that you're really excited about that you would like to highlight?
Well, we have, of course, other projects on these smart samplers that I'm excited about, but I wanted to share something else that we are also working on right now. It's a project that we are going to start. It is a project that is funded by the European Union, and it's this big project that it's not headed by us. It's headed by Professor Börje Sellgren from Malmö. So it's a large consortium consisting of 12 institutions and companies across Europe and also one in South Africa. And it will be a training network for young researchers. And we will train 12 PhD students in total in this network. So it is maybe a bit on the side on what I've been talking on earlier, like the smart samplers, but it still will have a focus on dry blood spot and other sampling techniques that can be done at home. But the overall aim of this project is to develop disruptive and greener technologies that can address inequalities in cancer healthcare, and also then to train these young researchers. So the project will use dry blood spots and similar microsampling techniques. We will use liquid chromatography, mass spectrometry, but it will be a different focus on other ways to extract biomarkers for cancer that are challenging to monitor.
Well, Trine, I think this sounds phenomenal. Really investing in and building the pipeline of expertise and early career talent in the field, I think is critical and something that the PCC heartily supports for anti-doping as well. So I appreciate you sharing this project. And now you've mentioned a couple of times now some of the ways that you work with the Norwegian Doping Control Laboratory. And I think these collaborations and kind of working with other stakeholders is key for advancing anti-doping. So could you maybe go into a little bit more detail about how you collaborate with others, whether it's individuals, labs, universities, these national anti-doping organizations and funding organizations like the PCC in the work that you do every day?
So we have research collaboration with relevant user groups. So that would be the Norwegian Laboratory for Doping Control. We also have some collaboration with Anti-Doping Norway. And since doping is only part of the application areas we focus on, we also have collaboration with clinical and laboratory environments at the Norwegian Hospital, in particular Oslo University Hospital. And then, of course, we have funding bodies that we are in contact because we apply for funding. So from organizations like PCC, which I've been so lucky that I got funding from, part of the project that we are working on with the smart samplers. And we also there was this project from the European Union that I mentioned. But we also work closely with innovation environments at the University of Oslo. We have this growth house and a tech transfer office that we collaborate with because we would like to move our research towards that line. And we also plan to involve users in upcoming projects, but we haven't done that so far.
Well, something to look forward to, it sounds like. It sounds like you work with a lot of different stakeholders, and you mentioned the PCC. I know you recently presented at the PCC's 2025 conference. So, Trine, can you tell us a little bit more about what your experience was like attending the meeting in New York City and sharing your work?
Yeah, this was my first time at the PCC conference because normally I go to these analytical chemistry conferences that either focuses on techniques like sample preparation or chromatography or application areas such as bioanalysis. What I found really nice with the PCC conference was that it was a conference where you had people with very different background that talked about the same topic from different angles. Like the common theme was doping, and then you had the scientists, and you had the government, and you had athletes. I liked that a lot, and I found it very instructive to learn more about the topics and see it from other side than only the analytical side.
Definitely. And did you get some good feedback on your work and maybe some ideas from people in the audience that you were able to talk to?
Yeah, I got some good feedback. So I will bring that back here to and see what we will do with that. So yeah, the opportunity to share the work to a broader audience than the analytical community was nice. We always want to try to find some niches where our research can be a useful tool. And I also enjoyed the venue and the city. So it's great to be in New York City. So I had a good time.
Well, I'm so glad to hear you enjoyed your PCC conference experience. And we've talked about some of the questions that you've been answering with your research and some of the phenomenal projects you've been working on. But I know oftentimes there are limitations, whether it's even just time, funding, staff, or the feasibility of the projects that you'd really like to do. So, Trine, if we took away all of the barriers that normally hold you back, is there one particular problem in anti-doping that you would want to solve most?
I think that was a very challenging question. But there are, of course, several things that are not in place and several things that could be highlighted. But I think that since my competence, so I will restrict myself, even if you say I don't have any restrictions.
It's easier that way.
So I will restrict myself to the analytical part with analysis and detection. So maybe it's not so visionary, but it's along the lines of my research field and dry blood spots and sampling. So I think that solving this problem with limited analytes that you can analyze by dry blood spots or microsampling, I think that will be very beneficial because it will reduce this burden then for the athlete and the doping control officer. You will take fewer samples and also you will reduce costs due to less shipping, and you can maybe ship it in room temperature and not on ice. And I think this will be relevant for both in and out of competition testing. There is a lot of research opportunities and things that need to be done for this to happen. And I think if you think even further, then maybe in a time far away, then there could be a potential of the out of competition testing being done in a more simplified way.
Definitely. And are there any particular analytes that you've kind of got your eye on next as targets that you think would be sort of the natural next step to move into being able to detect in DBS?
There are, of course, several potential analytes, but I think because our research group is not so big, so we try to keep a focus on the protein and peptide analytes. So it must be in that area, I think, that we maybe widen it a bit. And I would like to work more with some of these challenging peptides that are a bit smaller than HCG that we have been talking about now, but which I have some experience from now since the project that I presented in the conference. So these peptides that are like medium sized, they are difficult due to several reasons. So maybe broaden the field of analytes a bit that we normally work with. In a very small scale, I think that became that.
Well, I like that you put some of the bounds of realism on your dream project, Trine. I think there's so much to look forward to in the work that you are doing, and you've shared so much of your research with us today. If our listeners want to learn more, though, what is the best way for them to do so, or what is the best way to get in touch with you if they have questions?
Well, of course, the research papers you can find just by searching. Our research group have a homepage at the University of Oslo webpage that is called Smart Protein Analysis at UIO. And there you can read about and get an introduction to the research, like overall research and an overview of the papers. The publication list is updated, but the other text could maybe be updated a bit more. So if you want the newest news and not only the publications, then I try to share some updates on LinkedIn. So following my LinkedIn account, then you could get an idea of what our research group are working on at the moment.
Well, thank you for sharing all of those resources with us. Listeners, definitely take a moment to follow Trine on LinkedIn and take a moment to check out their university page and look up some of their papers to learn more about their recent work. And Trine, it's been such a pleasure to have you on the show today. Thank you so much for joining us.
It's been great to be here.
And listeners, thank you for joining us as well. If you haven't already subscribed to our podcast and left a rating or review, please take a moment to do so. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.